Alzheimer's disease is an enormous problem that, with an aging population, will only get bigger. More than 6 million Americans are living with Alzheimer's disease, and 1 in 3 seniors will die of it, according to the Alzheimer's Association. By 2050, the cost of Alzheimer's disease, currently estimated at $355 billion, will rise to $1 trillion. Could one of the causes of such a huge and costly problem be traced back to the cells that line the body's tiniest blood vessels? A new study published by a medical university of South Carolina, MUSC, research team in molecular therapy suggests that the answer is yes. The team, led by Hongkuan Fan, Ph.D., associate professor in the Department of Pathology and Laboratory Medicine, found fewer of these cells, known as pericytes, in the brains of people who died of Alzheimer's disease. They also found higher levels of FLI1, a protein most often found in blood cells and thought to govern their development. When the team blocked, or inhibited, the action of FLI1 in a mouse model of Alzheimer's disease, the memory of the mice improved. Blocking the protein also stopped immune cells from leaking into the brain and causing the inflammation that is a hallmark of Alzheimer's disease. Blocking FLI1 could be a promising new approach to treating Alzheimer's disease and other dementias. We are really excited by these data because they suggest that FLI1 could be a new therapeutic target for Alzheimer's disease, said Fan. Better therapies for Alzheimer's disease are urgently needed. It has long been known that people who have vascular issues, or problems with their hearts or blood vessels, are at increased risk of developing Alzheimer's disease and other dementias. These include people who have had a heart attack or who have diabetes or high blood pressure or cholesterol. That's not surprising, since the brain is hungry for oxygen. When it doesn't get enough, because the flow of blood is inadequate, its cells don't function as well and can begin to die. Lining the walls of tiny blood vessels known as capillaries, pericytes make sure the brain's energy and waste elimination demands are met. The capillary is where all the action is, said Perry Halushka, M.D., Ph.D., Distinguished University Professor of Cell and Molecular Pharmacology. It is the place where all these exchanges really take place. Pericytes also help to make up the blood-brain barrier that prevents impurities and immune cells in the blood from reaching the brain. This is especially true in the aging population, where vascular dementia is going to become a bigger problem. With funding from the South Carolina Clinical Translational Research Institute, the MUSC team looked at the brains of people who had died of Alzheimer's disease, drawing on the resources. The opportunity to study the human brain is an extraordinary asset for the institution and for the study of all types of brain diseases, not just Alzheimer's disease, said Halushka. The most exciting finding is that the FLI1 inhibitor actually improved cognitive deficits in the animal model because, in the end, that's the only thing that matters, said Halushka. The team also found that blocking FLI1 in the mice helped to prevent pericyte loss and preserve the integrity of the blood-brain barrier as well as reduce the buildup of amyloid beta. The next step for the MUSC team is to develop an RNA that could silence FLI1 and so reduce the brain inflammation that leads to cell death in Alzheimer's disease. The goal would not be to do away with FLI1, as it serves important roles in the body, but to maintain it at healthy levels. What's exciting is that this could be a new way to think about treating Alzheimer's disease, which has never been thought of before, said Halushka. This research opens up a whole new area for potential targets, not just FLI1 but the pericyte itself. Pengfei Li, Yan Wu, Eric D. Hamlet, Andrew J. Goodwin, Perry V. Halushka, Stephen L. Carroll, Meng Lu, Hongkuan Fan. Suppression of FLI1 protects against parasite loss and cognitive deficits in Alzheimer's disease. Molecular Therapy, 2022. doi. 10.1016-J. YMTHE. 2022. 01. 023 Medical University of South Carolina. Medical University of South Carolina. 2022, March 10. Could leaky blood vessels in the brain be a culprit in Alzheimer's disease? Science Daily. Retrieved March 10, 2022 from ift.tt/eapdinsk Medical University of South Carolina. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.